Hi everyone, welcome to Foresight's Biotech and Health Extension Seminars, sponsored by 100+ Capital. Really excited today to have Thomas Duffield here. And Thomas, you'll be discussing aging as a failure of epigenetic repair fidelity. Really exciting topic. I'm so glad so many people have joined for this. I'm going to be in the chat monitoring. I'm going to share a lot more info about you and your work in the chat so that people can follow along and get some background info. But I would say, without further ado, the stage is yours. Take it away, and I'll be in the chat until we hit the Q&A time in a little bit. So thanks a lot for joining. Really excited to have you on, and please take it away.
Thank you so much for having me. And I'll preface this by saying that Pedro Magalhães is in the chat. He's my supervisor. And Pedro's never seen me do this in anything less than 3 hours. So to do this in 30 minutes is going to be quite the feat. So I hope you're all keeping track of that, because this is like watching someone do high wire. Okay, so nice to meet you. My name's Thomas. I am interested in the root cause of aging. I don't care about the diseases of aging. I don't care about the drivers of aging. I don't care about the secondary consequences of aging and all this. I want to know what is going wrong right at the start. What is the very first thing? What is the thing that, as I discuss with you later, evolution can't do? And I realize that there's a debate about whether evolution can't do something or it won't do something, but I'm going to try and prove to you something that evolution physically cannot do. It is a logical impossibility. And then I'm going to lead you into how this causes a systemic problem that just is an unresolvable problem for dealing with noise in the epigenetic system. And that it's worth mentioning that this is something which is unique to complex organisms, and other organisms might have different age avoidance strategies. So just bear in mind, when I'm talking about this, I'm typically talking about mammals, things with an epigenetic system like one would imagine with a human. Plants and other creatures have different age avoidance strategies. Right, okay. So I'm going to assume that sorry, I forgot to share my screen. That would have been a good start. Everybody can see that? Wonderful.
Looks great.
Okay, so I'm going to assume most people in the audience here are familiar with what epigenetics is. I'm just going to run you through 2 seconds of this just to kind of like make sure we're all on the same page. Epigenetics is essentially the memory handler for DNA. Everybody knows DNA is a language. People have varying levels of understanding about what that exactly means. DNA is a self-executing bytecode. It differs from most other languages because rather than, say, a line of computer code, if you leave DNA to its own devices, it will run itself. And so when you're dealing with a language, you need something called a memory handler, which goes along and looks at where you want to start reading the language and where you want to end reading the language and which bits you're going to run. DNA has the exact opposite problem. DNA wants to stop everything running because it only wants to run the bits which make sense. And so therefore it needs to silence most of its genome. The way that it does this is basically with distance from DNA, access to DNA. So in picture A here, we have a nucleosome surrounded by DNA, and there is just physically no room, because it is so tightly wrapped around the nucleosome, for a enzyme or any kind of factor to reach in there and bind to the DNA and do something with DNA, typically access for transcription. If you reduce the amount of DNA methylation in this context, or you put some nucleosome modification in, like in C, you create more distance. And that basically is it. Later on, not so much discussed here, but that's the activation function of methylation. And that's how to think of that, I think. The activation function of epigenetics is essentially just one of distance. How much access can I give with modification? And then there are all kinds of different ways of controlling these modifications. You can have locally bound enzymes like in D, all kinds of different ways. But the point is that epigenetics is the memory handler for DNA, and epigenetics is controlled by distance. Quick caveat here. There's actually quite a few different I've heard a lot of different definitions of epigenetics. When I'm talking about epigenetics, I'm meaning something very specific. I'm meaning something which is inheritable, something which is inheritable into daughter cells, not into children. When the cells divide, you can inherit your epigenetic modification. I'm not talking about transient things like proteins that aren't going to be sent on into the daughter cells, where information is lost in between generations. So specifically, we're talking about DNA methylation, histone modification, histone variants, histone H1 variants, which is a massively looked at field, by the way, really fascinating stuff, and some types of RNA. But typically RNA, I wouldn't really include a lot of that in epigenetics because it's lost in between daughter cells. Right, so that's epigenetics. That's what it is, but this is the kind of the point that we need to lead to. If you take damage to something, damage being defined as when a system changes in a way which would be selected against by evolution. When epigenetics takes damage, when a region of epigenetics takes damage, and DNA methylation, I'm going to use DNA methylation as my primary example here, and I'm going to commonly refer to DNA methylation as epigenetics. It's because I get tangled up in the examples, but anything which is true of DNA methylation is also going to be true of histone modifications. When DNA methylation takes damage, you're really looking at information loss. You're looking at a piece of state information which has been lost. DNA methylations are arranged into internal gene CPGs and also CPG islands, which can also be internal sometimes. And these are the mechanisms by which these bits here, they are the bits which are controlling access. And when these go wrong, if I was to take one which was, say, for example, entirely methylated and then remove all of the methylations, that would be damage if it was incorrect. And that would result in information loss on the state of that methylation region, right? I've taken something which is being regulated, I've wrecked it, and now it doesn't know what it should be doing anymore. So the question is, how do we repair damage? There's only two ways of repairing damage. You can copy damage from backup. You can have an existing version of that entity and say, I know this is the right one, and this is the one which has taken damage, and I will copy from this to this. Alternatively, you can recreate something with an algorithm. You can recreate it through principle, is another way of saying it. You can say, I know this should be the case. And because this should be the case, I don't have to care about what its original state was. I am just going to set it to that state. I know that that is correct. What we're really talking about here is logical reversibility. We're talking about having a state that has taken on damage, and we're talking about returning it to a prior state. Now, logical reversibility has had some interesting maths done to it, and it's been demonstrated that there are all kinds of restrictions about the entropy of logical reversibility and how you can have entropy neutral logical reversibility and entropy positive logical reversibility, by which I specifically mean the ability to go backwards from a damaged state to its previous state. Sometimes you can do that, but sometimes you can't. And the states in which you can do that are states where you know that the type of damage, the thing you're looking at, that could only have come from one prior state. So if I don't know, chop a head off a teddy bear, and then I tell you the only thing that could possibly result in this teddy bear's severed head is a previously existing teddy bear, you can know that that's the state you need to return to make that logically reversible. But if I have something where it could have multiple options as to how that damage accrued, you can't go back in an entropically neutral way. It's impossible. This means that when you're dealing with these states, you're really trying to maximize your logical reversibility. I'm about to show you that state B is physically impossible in the human body. In fact, I'm about to show you state B is physically impossible, full stop. And it's not an inductive process. This is not something you need to prove with measurement. This is something you can prove with logic. If I have a backup of something, I have to have enough information in my backup of it that I can perfectly reproduce the thing that I'm backing up. That's not the only thing I need. I also need a way of telling the difference between my backup and my original. If I hand you two sheets of paper and I tell you there's an error on one of the sheets of paper and I don't tell you which one is which, you can't do anything with that. It's impossible. You will get it wrong 50% of the time. If I point at which paper has the error in, you can fix it 100%. This is an infinite regress problem if you are dealing with something which obligately has to be the outer layer to any system. This is the point. If I have epigenetics and I'm trying to make a backup of epigenetics, a system which is by definition a single point system with no way of comparing, because it's not like DNA. It doesn't have a phosphate backbone to provide trust. It doesn't have another strand of complicated dictionary legal characters. Epigenetics or methylation should be on or off. Those are the two legal states. And if I give you a hemimethylated state like an A, then there is no logical reversibility which is possible from just observing that single state. It could have come from two separate options. It could have come from fully methylated or no methylation whatsoever. So if I give you a state like that, and it has to be the outer layer, as I'm about to prove, I need to compare it against a backup. So I create another hypothetical set of methylation that tells me what that should be. Now, this doesn't exist in biology. I'm just using this to prove a point, just to bear with me here. If I do that, I need a system B to compare the two. I need to be able to tell which one is which. The problem is now B is my outer layer. And now I need some way of ensuring that noise doesn't enter in B. If I have an error in B, I need a way of being able to duplicate, make a backup of B, and then compare which one is which of that. And this is, as I said, an infinite regress. This will keep on going up infinitely. There's no way to not have this occur, which means you couldn't, it's physically impossible, design a flawless backup of the outer layer of any kind of information structure, in this case, epigenetics. All you can do is you can try to mitigate the damage. Now, some clever people in the audience are inevitably going to start saying, what if you compress it? What if you have an original and then you have a backup which is compressed? The second you start compressing it, you start dealing with a system of logic for handling the compression of that information, which means you've moved into the algorithm version of this, using an algorithm which defines entire sections of methylation, entire sections of epigenetics according to a system of rules. Turn this entire CpG island on, turn this entire CpG island off. If this CpG island is below 30% methylated, fully methylated. Things like that. And the problem with algorithms is they have a fidelity problem. We have, I think it's about 30,000 CpG islands in our body. And each one of those contains several thousand individual CpG entities. If we ignore every entity and we just treat every CpG island as its own thing, that's still a crypto key that's with a binary state. And we assume that there's only a binary state. It's either off or on. There's no states in between, which, by the way, isn't true. Never any in-between states are observed. We've got a crypto key with 30,000 positions. I don't know if anybody's familiar with how that math works out, but that gets very, very hard to calculate very, very, very fast. So you end up in a situation where when you're designing one of these algorithms, you cannot achieve perfect fidelity of data reconstruction. It is not possible, given a certain size of algorithm and a certain size of data, to which DNA and epigenetics massively surpasses, to get to the state where you have enough fidelity to flawlessly reproduce your information when it accrues damage. So, and the more information you can spend on the size of that algorithm, the more fidelity you're going to get at the end of the day. Let me give you an example. If I had an entire second genome and all it was for was designing a complicated protein algorithm which went around and worked out what the methylation state of the first genome was supposed to be doing, an incredibly complicated algorithm, I could potentially get a high degree of fidelity there. But how do I say this? But then I've got a secondary problem of data blowout for a start. But also, if I have a smaller algorithm, if I can't afford to double the size of my genome, then I have to start making even more compromises. And eventually I'll get to the point where I'm dealing with lossy compression. So what I think is happening is roughly something like this. We've got no backup to epigenetic information. We've got an algorithm which is restricted below perfect fidelity that isn't capable, when damage occurs in the epigenetic systems, of flawlessly recovering that damage. I think that that's broadly completely not a problem for very simple organisms, because they can encode the majority of this information in DNA, and DNA has a trust system. It has an outer layer which is capable of just preserving that, and it can basically be explicit and hard-coded with things. It can just run the entirety of its DNA, and it can encode all of the important parts in something which is explicit and doesn't require any contextual modification, doesn't require state to be retained in itself. And that's going to be really important in a moment. But the second you start moving into epigenetics, you start requiring systems which retain their state within themselves, because the only way of storing that much information, due to the fact that it's the outer layer and due to the fact that it's an enormous amount of information, is you start having to, rather than be able to explicitly tell something what it is, you ask it what it's currently doing, and then you make a best guess as to its state and its logical reversibility from that. And then in the extreme version of this, I think that what happens is you have the cellular context providing the information to tell the epigenetic system or tell the cell what it should be doing. So things coded for by DNA, like housekeeping genes, they fall into category 1. Things which involves the majority of the human cell, which is controlled by epigenetics, fall into category 2. And things like cellular differentiation, which require so much information that it can't be stored in even epigenetics, is stored in the context of the local environment. And I think this basically means that when you've got a simple organism, you can achieve lossless compression, which means you can go backwards. That basically almost reads as logically reversible compression. You can have a bacteria, and basically whatever its proteins are doing, you can always work out what its state should be, because you can just go and read its DNA, because the answer is its DNA should be running at maximum capacity, producing as much of this protein as possible, regardless of the circumstance, right? But when you start moving into these forms of information storage in the cell, you start dealing with lossy compression. You start making trade-offs. You start saying, it's probably worth me not making this a perfect repair job in the name of me not having to expend a massive amount of genetic data. And it's worth pointing out that every piece of genetic data, every byte of DNA or anything you add to a system gives it more potential for error. There is a selection against extending the genome, a selection against extending epigenetics, and definitely a selection against extending context. And I think this is basically what the difference between a simple organism, a medium organism, and a complex organism is. I think a simple organism is an organism which can essentially store all of its information in DNA. A medium organism is an organism which most of its systems or some of its systems are epigenetics free, and then a few complicated ones which need to respond to its environment, like in C. elegans, they are epigenetically controlled. I think that these are, on the way, incredibly bad models of age. And then complex organisms like us, we have such a complicated body. We have such a complicated system of responses and cell types to thousands of different situations. We are such a intricately designed citadel that we require so much information. We go past this fidelity limit, and then we go past this fidelity limit as well. And you just can't store all of this information in epigenetics anymore. You start having to store some of this stuff in context. And that's where we start getting into, I think there should be a slide, self-referential identity. Self-referential identity is when I define myself. I am because I am, right? And the problem with this is the fact that if I change that identity, I introduce noise into this identity, and I've been basically forced by this fidelity limit to encode my identity in the context of what I already am, so to speak, then when I take on damage, I'm not just taking on damage, I'm taking on my ability to detect damage. I'm taking on a decreased ability to reverse myself into my previous correct state. And we'll come back to this slide in a moment. What I think is basically happening is that the regulation of epigenetics is not just information loss, because information loss happens across the body, across every cell. There's tons of information loss in all circumstances. Proteins are misfolded, DNA acquires mutation, the DNA damage. The cell is a noisy place. It's not the information loss which is a problem. What I think is that the self-referential identity of elements which regulate epigenetics, or another way of saying it, the epigenetic regulation of epigenetics has this self-referential relationship, which means when noise is acquired in it, it can't fix itself, and you get a feedback cycle. You get a problem where its ability to fix itself is eroded by the damage, which prevents the next piece of damage from being fixed as well as it previously could. And you essentially collapse into perfect logical irreversibility. And this, of course, because these systems don't just regulate themselves, but they regulate other systems, as these collapse in their ability to regulate transcription, so do all of the other systems. They regulate collapse. And as far as what I would argue is that gives the phenotype of age. So that's the theory. I got really good results. That's a graph of how noise accumulation in CPG loci and CGI, that's CGI, how noise accumulation in these correlates to age. As far as I'm aware, I don't think anyone's ever got a correlation like that between anything and age. That is strictly a 1 on that graph on the x-axis, represents a perfect correlation between your increasing age and the variance within your CGI loci. This is from a data set of 1,400 samples, 25 per year between the ages of 30 and 85. Multiple tissues. There's brain, lung, adipose, blood, white blood. And I've done it in the individual tissues as well. We'll get to that slide in a second. But that's incredibly high correlation between the spread of how these CGI differ from one another with age and age itself. If to get a high score in this, to reach 1, as your if I take a bin sample of a year group, and I do this progressively for a 25, and I get the standard deviation of each group, and I track how that standard deviation increases with age, for this to be 1, it has to perfectly increase with age. And so the majority of CPGs do, in fact, show this behavior. There's this peak here, which is very interesting, isn't it? And a better way of looking at this is to look at the individual tissues, because the different tissues have different populations of CGI. And while some things are retained between them, when you start looking at them, you start seeing the 3 characteristic peaks, because this one is so strongly positive, it actually swamps one of the peaks. So let me walk you through these graphs, because they're kind of the point. This peak here, these are the typical CGI. They aren't, actually, I think these are CPG, either way. These aren't as far right shifted in these graphs because of the binning, and because these are 5-year age bins and far less samples, because the data set which produced this was a combination data set of 7 other EPIC data sets. These have far less samples, and so then to do fair comparison, I have to restrict the age bins between the to the lowest common denominator number. So that peak is the same peak as that peak. That peak there is the same peak as that peak. And that peak there is hidden in this. And what we're seeing here is that there's this typical CGI population, typical CPG population of the typical amount of correlation you get with age. Here, very noisy again, because I had to bin into such large age groups and had so few samples. And here, there's this second peak that's much, much smaller. If you can imagine that one going up and then coming down, you can see that the actual amount of these of entities in this peak is actually very small, but consistently seen, by the way, in all of the data sets which I just looked at. And I think these represent the self-regulating CGI, the ones for the regulation of epigenetics. And then here we have the age-responsive CGI, the CGI that change with evolution in response to age. So evolution is switching something on in response to age, which results in a decrease in the deviation of a CGI island because it's had a pressure applied to it, which I won't go into much detail now, but it's in the paper. And this is characteristic. You'll always see this peak when you look at this kind of thing, and you'll always see this peak here, and this peak not in blood so often, but it depends. And this is very high correlation when you do it with high fidelity in large amount of samples, and very distinct features. Those 3 peaks really do seem to be the 3 classes of NAC, or noise-associated correlation, that we see in all the data sets. So what I did was I got a standard neural network classifier, and I did what we called variance isolation preprocessing. So what this is very simply is just you take a CPG feature or CGI feature, you rank it according to age as standard, and then you take the results and you give them a rank. So the lowest result is rank 0, the next result is rank 1, 2, 3, and so forth. And what you're trying to do is you're trying to eliminate all evidence of beta values, all evidence of the actual state of the methylation loci in question, of the CGI in question. And what you're really caring about is the relative dispersion within that CGI compared to the rest of the population within that column. And when we do this, we get neural networks which can predict, I think that's sub 1 year, which I actually don't trust because the ages are commonly reported in 1 year integers. So I think that most of the time when we get that particular class like that, we think it might be a little bit of variance. But generally speaking, we always manage to get incredibly good predictions. I think the average across all data sets is 1.74 years. But again, this is made with a epigenetic clock. This VIP clock is made with no information of beta values whatsoever beyond the relative dispersal of the loci. And that on the right is a single CPG clock. And we've made hundreds of these, and they're all the same. They all are predictable with the exact same processing. You can take a single CPG loci, anywhere from the top, like this, like top, what I'd say, like quarter of the graph, quarter of the range, about 0.5. And you can make it, it's predicted for age. And so, by the way, this is what I think Horvath's clock is detecting. I think Horvath's clock's detecting this. And then what some clocks, some blind clocks are detecting is a combination of this and this. And what we're trying to detect based off is just the noise, just how much this correlates. So by the way, just as a quick side note, I think there are two types of epigenetic clocks. I think there's AR, age-responsive epigenetic clocks, and there's self-referential epigenetic clocks and general noise clocks. So the fact that we can make very predictive single CPG clocks out of this is quite encouraging as well, because this really should be removing all of the information that a traditional clock is using. And this is the punchline. If you take this peak, just these samples, and you work out what they enrich for, it's pretty much the regulation of regulation. It's pretty much exactly what we thought it was going to be. It is the things which, when they go wrong, they control themselves. Epigenetic regulation of epigenetics. And that represents the class that is the highest group, the highest, most correlated group that is a distinct entity of these CGI. And so broadly speaking, what I think is happening, I'll just leave it there for a second. What I think is happening is this. These self-referential CGI, the ones which do this, and are represented here, are going wrong when they acquire damage that they can't deal with, because the damage which they acquire makes them logically irreversible to themselves. This is a feedback cycle which gets progressively worse across your life. It will start slow and it will stay very small and slow for a very long period of time. But like all feedback cycles, it will have exponential growth. Then you'll have progressively more overflow into the rest of the systems, the other systems which are regulated. Put another way, this peak here, represented by the majority case here. This majority case is pretty much this, mostly. And this together is going to result in the phenotype of age. And it's attractive, not least because it fits perfectly what we see with age. We'd expect to have a period of no visible damage, and then the majority lifespan period of not much damage, and then a dramatic collapse at the end as the network fails, as the exponential damage, the exponential erosion of epigenetic identity in the self-referential systems leads to the regulated systems becoming crushed. And I don't know how I'm doing for time. Is that pretty good? Wow, that's really close, actually. Before I finish, I'll just simply say that I've had to rush through a lot of arguments here. As I said, this normally takes 3 hours. And so there's going to be a lot of caveats, a lot of things where you're going to say, what about this? And we got a draft up on bioRxiv, but some of the really good results aren't even in that yet. But the arguments are all going to be there. Yeah, and so that's about it. So just to summarize, basically what I think is happening is this. There's a class of CPG that's unique, SRCGIs, SRCPGs, and they take on noise. Backup is logically impossible. Mirrored backup is logically impossible. You have to use an algorithm, and compression is a necessity of that algorithm. The amount of information in a complex organism is so high, it demands a lossy compression. There's a fidelity limit that makes perfect fidelity impossible above a certain amount of information necessary to create or control an organism. And therefore, because you're using lossy compression, you will take on noise, which is correctable in most systems, but not those that are self-referentially defined. This will disrupt their identity and their ability to preserve their identity in a feedback loop. And this disruption of their identity preserves the identity of all of the other epigenetic elements. They are the definers of them. And so when that fails, it's a systematic disruption of repair fidelity across the genome, and that's the progression of age and the phenotype of age. Essentially stemming from an impossible system at the top of biology, something evolution cannot design. Thank you very much. Thanks for that.
Wow, that was so fast. And you already have a lot of different questions and hand raises. I would say we go with Creon first, then we'll be moving on to Cosmo, and we have another hand raise, and then we go with Micah and Monte. Does that sound good for a queue for now? And then I'm sure that there's plenty of more questions that will topple in. So maybe Creon, you go first.
Okay, thanks, Alison. Sorry, it took me a sec to unmute. Thanks for a very interesting presentation and for your really profound connections between reversibility and this kind of damage repair stuff. it was a little fast and a little abstract for me. So I have some questions that might help clarify things. And Alison will probably have to cut me off because I have about 3. So my first question is, so you seem to have this infinite regress argument that you make about backups, but on computer systems, we make backups and we don't expect them to be perfect. But basically, when you make a backup with a computer system, and then I'm just being informal here, you copy some files or some disks and you make another copy and you put it in a vault. And what I mean by that is you don't access it all the time. Its only purpose is an emergency restoration purpose. So you don't access the files every day where they can get corrupted and damaged either by human activity or failures in the hardware. You copy the stuff. It used to be on tape, and you just stick it in a room, and it's not going to get damaged, or excuse me, it's going to be much less likely to be damaged than the day-to-day copies that you're using. Now, so we don't expect it to be 100% reliable, but we expect to get a huge amount of assurance that we can restore a likely correct copy if necessary. So my question is, why do we care that it's like theoretically slightly imperfect when we make backups, whether it's in organisms or in computers? How does your central idea differ from the idea of backups on computers, meaning they're good, but they're not perfect? Isn't DNA itself a backup with its double-stranded and it's all wound up and protected? And it is, again, it's only exposed and single-stranded and copied during transcription. And then RNA is used as the working memory with the ribosomes reading it and amplifying it. So it seems to me like we already have a backup hierarchy in biology, but are you saying it's insufficient? And then I've probably gone on long enough, but I have one more. What put as a separate?
Before, can you ask that in a second, because there are 2 separate questions really there, and I think that my brain will melt. So the first one is I don't think there's this backup. I don't think this backup exists. I don't think you can have a perfect backup. That's the point. It's a thought experiment to demonstrate that a perfect backup isn't possible. So you have to deal with an algorithmic backup, right? You could call it a mirror backup versus an algorithm backup. And yes, hold on. But yes, the next thing is that a certain amount of noise would be theoretically tolerable in some situations, because you can design systems so that they can tolerate noise, and as long as the noise can be reversed in some way, that's acceptable. But so that's the comment on there. And then, sorry, I'm already losing the second question.
That's okay. You've addressed it. I said, isn't DNA itself kind of a backup, at least with respect to?
Oh, right. Yeah. So DNA is a bit more complicated than I think people commonly conceive of, at least in the layman, because DNA has a trust system built into it. It's a phosphate backbone. That's what I think. I think that the phosphate backbone basically is a trust system, which allows for the DNA phosphate backbone, the sugar backbone. So the things the bases are connected to, the thing which breaks in most cases of damage. That's essentially a trust system telling you which strand of the DNA to use as a template for the other one. It's saying which one. It's a built-in kind of like B or C there. It's a built-in way of doing that. And it's possible because it does have flaws. And there is a version of this where it does take on damage, right? If that's the system, then what would be the version of noise in that system? It'd be a situation where it's logically reversible within that state, i.e. double strand break. And so, by the way, another the word I think we should really start using to describe epigenetic damage is epigenetic mutation, because epigenetic mutation is more kin DNA mutation, where you have 2 dictionary legal characters that have been replaced because there's no logical reversibility. A choice had to be made and it's the wrong choice. And so you ended up with a silent, unrecognizable change within the scope of the immediate repair enzymes that would require external context information, which is unavailable or impossible to kind of like.
Right. No, I fully follow. I fully follow your reversibility versus irreversibility arguments. It's quite nice. Last question: isn't there a reset for all this stuff? And this reminds me to some extent of the work of David Sinclair. Like when a new baby is made, it's not old, it's young. So somehow there's a reset.
Why we become young is the most important question in aging research. I can't conceive of how we haven't spent the last several hundred years constantly asking ourselves this, right? yes, that's a reset point. What is lost when you've got two adult old tissues and they come together and they make a new young tissue? The fact that there's epigenetic stripping in the primordial cell seems just the most obvious possible answer to that. But there's an interesting caveat there as well. You're trying to get back to what you're really trying to do with childbirth is get back to a logically reversible state. You're shedding information until you reach logical reversibility. You can do that with a stem cell, but only within the context of a certain amount of failure. So I think what's happening is when you've got something which is an error which is occurring, let's just take a non-epigenetic example. You've got DNA damage, right? A repair enzyme comes along, it tries to work out within the context of its immediate scope, the things that it can see, can I be logically reversible here? It finds a bulky adduct on one strand, not the other strand. That's an illegal character on one strand, a legal character on the other strand. Logical reversibility collapses into a repair decision, right? If you have a problem like a double strand break, which gives rise to cancer, and the cell itself can't be worked out, then you've got a detection mechanism for that cell being incorrect. And then you've got a problem, how do I replace the entire cell? Again, it's logical reversibility. You go back to the stem cell. Now, the stem cell is your point of known context. You're the point where you can determine that whatever type of damage this is where it came from. And then you can build out from there again. And the most extreme version of that is childbirth. I can't help but see now childbirth as the necessity to create children, as the shedding of all of the information unnecessary to the purest signal in a biological organism, an age shedding strategy, a noise shedding strategy.
All right, we'll leave it at that because we have so many more. And next one up, we have Cosmo.
So it sounds like you've effectively discovered the aging equivalent of what we would call heteroplasmy with like mitochondrial mutations. That being said, this noise metric is going to give you a different set of genes that are most strongly correlated, right? Like the genes that you would specifically choose to make a better clock. I'm curious as to what pathways emerge when you do that analysis.
So these ones, and others, I just picked the top, whatever. They're the regulation of regulation. They're the things involved with binding histone modifications, binding DNA at specific locations so that you can bring epigenetic change. All of the things that if this goes wrong, it's going to affect itself epigenetically, right? So, for example, if I have something which binds DNA at a specific location, rips off a histone that is necessary for silencing a gene, that's actually going to affect itself because that gene is probably controlled by epigenetics. And that's what we see.
Yep.
They're self-regulated CGI. So.
I work at the intersection between obesity and crosstalk between DNA methylation and RNA methylation. So we should definitely connect offline because I think there's a lot of overlap here.
Sounds good. Always fun.
All right. Are we happy or can we move on to Micah? Great. Cool. Micah.
So I have a very quick question just to get a little clarity and then my actual question. In the noise correlation graphs, what exactly is that noise? Is that just a difference of methylation between various cells? Like you have a bucket of cells. Is the noise the difference in methylation status between all of them, or is it something else?
So specifically for each CPG site, each loci, each CG, that's one feature. For every year, 25 samples in this particular graph. So 25 individuals of that year of age. So 25 35-year-olds, 25 36-year-olds. Within that 25 individuals, take the standard deviation, and then that results in a column of standard deviations according to the year of age. And then does that column correlate to the increasing age? And somebody is inevitably going to mention measures of entropy here. And I spent 6 months dealing with the measures of entropy. And the problem is this actually turns out to be really the best way of handling that. I think a lot of people dislike it because they think of standard deviation as simplistic. But when you're dealing with measures of entropy, you actually miss a lot of the information or rather, it's very hard to display it to a human because you end up measuring entropy in very specific subscopes of what's actually happening. And then you have to aggregate it in a very complicated way, which is very human arbitrary as well.
Okay. And then for the correlation chart, that one of the first correlation chart looked like it had a correlation of basically 0.99, right?
Yep.
That feels way too strong of a correlation to be stochastic. That feels like a very intentional clock. There is very little in the world with a correlation that tight unless it is designed to match.
I think it's causal.
Like, year by year. Like, it feels like if this is truly a stochastic process, like random mutations or random damage or random mistakes, I feel like you would not hit a correlation that perfect to chronological, like, number of times around
Unless
the
you're
sun.
measuring the same thing. Unless age is the same thing as deviation of...
Yeah, so I can definitely accept that. I think what I'm arguing here is that it feels like the cause of that cannot be stochastic. It must be intentional to get that type of a correlation. Like those
what do you mean by
changes in
intentional? I'm a little bit...
intentional in the sense that, like, evolution decided that it wanted to track how many times around the sun we've had, and it uses this mechanism to do that.
I think that the normal distribution here is really representing that stochasticness, the stochasticity. There's going to be some where by fluke it doesn't quite fit as well. But if it is the general rule is that as these things spread, they spread in a way which correlates with age. You're going to see. I see no reason to justify that with engineered senescence. To be honest, I think that if I'm measuring something and I'm measuring one thing and it's the same thing as the other thing I'm correlating it to, I should see a normal distribution centered on exactly one. And that's pretty much what I'm seeing.
Right, but the one thing you're measuring against is number of times around the sun, right? Which seems definitely not the same thing as methylation.
No, but I'm really measuring at I see what you're getting at. Right. So what you're saying is that humans have very different lives, and humans therefore have very different, like, what happens if you live next to Chernobyl, the mutation argument, right? Like, you receive thousands and thousands of times the mutation of somebody else. Right. With you. Right. Sorry. The reason that I don't think that happens is because I think that essentially the self-referencing CGIs essentially act as a rate limiting threshold. And so it's essentially rate limited in everybody. So if you have a massive amount above in a short amount of time of system collapse, it's detectable in a way because it's so unique compared to its compatriots. More of that argument actually ends up in the paper. That's a really complicated argument, but I basically think that we're rate limited, but we're threshold limited by the self-regulating CGI.
Okay, I'll definitely read the paper. I have many more questions, but I will let other people go, and if we still have time, I'll ask more.
Thank you.
Wonderful. Next one up, we have Larry.
Yeah, hi. I basically had the same questions that Mika had. What do you mean by measuring noise and methylation, how it's actually being measured? Usually noise is the absence to detect a signal. And so I don't know if you're looking at different sites and you can't tell what's methylated and what's not methylated. Is that what you mean by noise? And also aging to me is more of a program. There's some internal program that most, particularly mammalians, have, that when you go from the 8 cell up till, if you live to 100 or something, there's a program. You lose your thymus at a certain point. You go through puberty at a certain point. You can accelerate that, like progeria, when you have a mutation in lamin, which holds the nucleus together, and it does affect gene expression and things like that. So I'm just not sure. Certainly damage to DNA can activate or can inactivate P53, which would then lead to a lot of mutations and probably lead to cancer. And foam cells, of course, if they get damaged, they can start getting bigger and eventually probably cause you a heart attack. But to me, aging is more of some program that is innate in a sense in most mammalians, and how to interfere with that. I don't believe it's just noise. I think it's some internal program that we don't fully understand, because biology is really complex and there's a lot of different systems and things like that. Yeah.
So I think that's your program. I think that one of the things about the study of age, which is really important, and I skipped a load. There's some slides I took out of this because I didn't want to bore you with process, but the most important thing of studying age, and I'm a very, very big cheerleader for this, is definition. Definition is arbitrary. You can define something, and it's a statement that is a entity that exists within an idea that you can then look for. You can try and observe it in the real world, and maybe you see it, maybe you don't. But when people are talking about age, if you talk to 5 different aging scientists, you'll find 27 different definitions of age. And none of them are more or less right than the other. They're just really using the same word for 27 different meanings. And so when I'm talking about age, I'm not talking about development. I would argue that what you're using the word age to mean is development. Like in fact, I would argue that in some regards, you're combining the word development with some elements of what I would use the word age for. When I'm talking about age, I'm talking about cellular static breakdown. I'm talking about something which evolution would ideally not like in most situations. There's something where there is a systematic failure of the cellular network, the cellular system, in a way which results in ultimate organism failure if it's left unaccounted for. And I don't think that I'm not making any claims about development at all. I'm not saying it doesn't exist. I'm not saying that I'm measuring it even. I'm just saying that's development to me and not age. That peak there, that's, I think, the genes which are being switched on in response to age as part of a program. I think that this is, you have a decrease in noise with age, it's in the negative reaches, because a pressure is being placed on a CGI to turn on or off a gene, which makes you better at handling age. And that evolution can select for that. And so I fully believe that that kind of stuff exists. I just don't think it represents the majority of it. And the reason I don't think that the complexity of something is necessarily problematic or is because aging is a continuous process. There's an enormous amount of things happening, but you can produce a steady gradient with something with lots and lots of small things. And what I'm arguing is here, lots of CPGs are going wrong in different ways. They're some of them going up, some of them are going down. But that's resulting in is a steady rise in dysfunction, which is what ultimately results in this kind of ubiquitous, homogenized profile of age, I would argue. But does that answer the question, I think, maybe?
Yeah, I'm still not sure, again, what you really mean by noise and things like that. What's the actual physical measurement of noise that you're actually, what's the experiment that you're measuring epigenetic noise? Epigenetics, acetylation probably plays a bigger role than methylation of histones and things like that. And there's a lot of different control mechanisms, and there is crosstalk between methylation and acetylation of DNA, but it's just epigenetics is a real complicated phenomena, and we can measure DNA methylation easy. It's much more difficult to measure what's going on with histones and things like that. Yeah.
So there's a slide I removed from this, and I realize we've got to go past this question for time, but there's a slide I removed from this. It is in the paper, basically making the case that the methylation and frankly tied to histone modification is the summarizing function of the cell. It takes all of the various signals coming from everywhere else in the cell and it brings it down to one number. How much should this gene be produced? Because something has to at the end. No matter how complicated and a myriad all the different signals are, sooner or later you have to make a choice about whether you're going to produce that gene or not. And I produced some graphs. I'm very proud of them. They're a bit not necessary for the full flow of this, but they are in the paper. And basically I'm making the case that there are correct outputs from methylation, aggregate states. So if you've got a function, which is an activation function, you're bringing all these signals in, there's only a few defined outputs, which makes sense for evolution. And right at the end of this talk, I'll dig out the graph because it's really clear cut. That's exactly what I found. I'm very proud of it, but it's going to be its own separate paper, I think, slightly different from this. Because it's not just age relevant. I think that basically methylation is looked at wrong.
Next one.
Okay, great. Thanks, Thomas. Yeah.
Wonderful. We have 2 more questions to go to. Let's see if we can make it. We have Monte next.
Okay, can you hear me? I'm not sure if my mic works.
Yep.
All right, I'm just still a little weak on my information theory. Aside from reading Claude Shannon's paper, I was wondering what you just recommend for me to help fill in that gap for my own knowledge.
It was a good start, but it's basically set theory. That's what this all kind of essentially boils up to. So basically, if you're mathematically inclined, anything touched by von Neumann, and then he made it very accessible because he was a great speaker and a great talker about it. But you don't need to go quite so far. You do need to just understand the different versions of entropies and the different ways that you can. I'm not sure what a good answer to that question is. I just learned by doing, shall we say. So I guess just find an interesting problem and try to analyze it. That's what I'd say.
Okay. I've had a similar insights with set theory. I just haven't connected the dots, but you just did that now. So thank you.
Cool. Last but not least, we have Greg. Hey, if you want to ask a question, or I can ask it if you can't.
Hi, I can ask it in a couple different ways. So the question that I typed in is that I'm sure you must be aware of David Sinclair's experiments recently in which he claims that You can reverse aging, and your theory says aging is not reversible. So how do you, the first question is, how do you deal with that?
That's not what I'm saying. I'm saying the body can't reverse it without shedding the entire organism. So
That's not within the scope of Sinclair's experiments. So you have to address Sinclair's results.
So first of all, I think the first person to come up with those results was Campo with his age amelioration in mice paper.
Sure, that works.
And that's not inconsistent at all. I'm not claiming that you couldn't alleviate age in this. I'm just simply saying that the body can't, because it doesn't have the relevant contextual information. I can't really comment about the partial reprogramming stuff because I'm pretty sure I know how partial reprogramming works, and I'm actually writing a paper on that as well. So I can't. My argument for that is essentially unpublished research. So is that kind of? Can I just simply say, watch this space in about 3 months' time? Is that fair?
Yeah, that's fair, but just understand that both methods of reversing aging are contradictory to the idea that the body doesn't know how to do it. Along the lines that were being discussed before about aging programs,
I can't answer this question, and I desperately want to, but that's not quite true. So the body can obviously do it, right? Let's say it this way. Primordial cell generation obviously reverses age, right? It strips away the epigenetic modifications, and that happens. How partial reprogramming is doing that without losing identity is an interesting subject matter. And again, watch this space. But I'm not saying that the body can do it in no context. I'm saying the body can't do it in the context of an adult, complicated organism because it doesn't have a fixed point on the map for these systems.
Right, but that theory...
but it does have a fixed point on the map when you go down to stem cells. Stem cells being essentially the default ground zero of any particular system.
But I would say that there is essentially a map that defines every age in terms of what's going on genetically at that age, and that is what the epigenetic aging clocks are picking up on. And you say it's nothing but noise, but there's far more going on than just noise.
I'm saying that Horvath's clock is probably measuring mostly this peak, but maybe some of this peak. But the DNAM clocks that I forget the author, somebody will help me out, 1,000 CPGs, untrained, unsupervised beyond age, just pointed at this. I think it's picking up both of these. So I think that it depends on the class of clock. I think there's two distinct versions of clocks which pick up noise and clocks which pick up intentional change with age, selected for change in gene expression.
Right. There's so much to discuss here. There's no possibility that we're going to hash out all of the questions that we have.
Welcome to my hell.
Right. Right.
I have a paper on bioarchive which covers a lot of stuff, and there's more coming. So
I just want to refer to a couple things. So are you familiar with Leslie Orgel's error catastrophe theory?
No. Not at all.
So Leslie Orgel had an argument very similar to yours, that aging would be caused by a failure of repair mechanisms. And as that progressed, the repair become less and less fidelitous, if you can use that word. And eventually we would end up with a bunch of random mutations in proteins. And the theory is a complete failure. It just doesn't work that way. But what does happen is at a certain age, you begin to withdraw DNA repair deliberately through program mechanism. You begin to withdraw, say, for example, coenzyme Q10 synthesis in cardiac muscle cells. And of course, there are downstream sequelae of those things, but they're not random events. It's not that the body could avoid those things. It chooses not to avoid those things.
I would argue that's much akin to an engine rusting and then you deciding to not turn on the bits of the engine which might explode if you use them.
You can argue that, but you can't prove it. And the other thing is that because the program runs at different speeds in different people, we already know that different people age at different rates. And you're lumping in your argument here people that age rapidly from people that age slowly. And then you're saying, oh, look, there's chaos increasing in the system because there's less uniformity within a population. But of course, we already know that to begin with. So I think that what you need to do, and the question was really raised earlier to you, is to define the meaning of this noise that you're looking at. Is what's the functional significance of these changes that you're seeing? Are they sufficient to explain the pathologies of aging, Or are they just something that's happening that is going along with
So-
aging but doesn't really matter?
So I apologize if I've been unclear. If you have a CGI region and that CGI region has a correct state, it should be off. And I
Well,
systematically...
but there's no such thing. It should be off at a certain age and on at a different age. And it should be on when a certain other one is on and off when a certain other one is off.
There is
So-
such a thing. If you take a specific time point within a specific context, there is a best case for survival of the organism for that CGI state. Right?
If you're
Best
12
case scenario.
So
Yeah.
the best case scenario for survival of an organism is being 12. And if you're younger than that, you have a higher mortality rate. And if you're older than that, you have a higher mortality rate as well. So we don't define aging that way. We define aging as what happens sometime after maturation. And so there is no one reference state that defines optimum viability of an organism, because when you're 20, you're called upon to do different things than when you're 12. And so you must
Right,
have
but those are two
gene
different time
expression.
points. That's
That's
what I'm saying.
right.
Within one context of a time point, within one specific moment, there is a best case scenario for an organism. Saying that's different when they're in a different time
Yes.
point. Yes, that's a different definition.
Yes.
So I don't see why
you're-
that's problematic.
so
Anyway,
but
Maybe we
how-
should continue this.
Yeah, well, that's the problem. We just can't have enough time here. But it goes back to the definition of noise, and you have to define noise as departure from the ideal at any given age. And I'm not sure that you've done that. And I may be
Well,
wrong. Maybe
That's basically, yes, but that's what I am specifically defining it as, that you have a specific state, which is the optimum situation for the organism, or the best it can achieve, which basically is the best, right? And then anything which is a stochastic process, or anything which is introducing error into that, is damage. And the accumulation of damage within that is, what's happening is that gene, that whatever, it's a signal. It's trying to send a piece of information further up the line. And that signal is failing because elements within it, dictionary elements of the signal, are failing at that specific point in time due to the damage. That's what I'm
Well,
arguing noise is
I'll let
specifically.
Greg
But
I'll
we've
let
got,
Greg
yeah, we've got to
on
stop.
for one
Sorry.
second, and then we're going to wrap it up by 5 minutes after. Greg, do you have any final words on this?
yes, I do, but I think I'll spare everybody and let you move on to the next questioner. I don't want to monopolize the discussion, but I think there's a lot of assumptions built into this model that don't necessarily line up with what's actually happening in the real world. But I don't understand the model well enough, so we'll just have to move on.
How about, because we're now already 5 minutes over time, maybe Thomas, you send us any other further infos for other talks, because there's so much interest, I think, in digging deeper into this. And then we're going to share it when we share your recording with the rest of the group. Do you have any final words and how people can find out more about your work and how people can collaborate with you? Any final bits and pieces that you want others to know about before we close out?
So I have a paper on bioarchive. Hopefully that will be updated in the very near future with these results. It actually hasn't been updated with that specific graph that's on the screen. Always interested in people with interesting concepts within this space. Please recognize I did this in 30 minutes and 30 minutes of questions, and this has a lot of caveats that need to be addressed. And I hope the majority of them in the paper, but there are some which I think still need to be addressed, in the sense of I have the answers, but I still need to stick them in the paper somewhere. Because there's unfortunately word limits, which work the same thing as time limits. But my final kind of comment is, feel free to fight me. Strongly spoken opposition is obviously wonderful. Criticism is a necessary part of this. So if you think I'm wrong, please challenge me in any way, shape or form that you feel. There's emails, or just contact me and we can arrange a meeting. Any kind of way of criticizing this, I'm beyond interested. I'm grateful. So yeah, thank you so much.
Wonderful. Thank you so much for joining. This was really, really, really fun. And yes, maybe we have to have in the future a 3-hour-long seminar again, like we did during COVID a few times. Thank you so much, Thomas. Thanks for all of your amazing questions. This is a really fantastic group. And as always, really, really fun to host these. Thanks, everyone. I hope to see you at the next one. Have a good rest of your day.